All right. Hi, everyone. My name is Brian McWilliams. I'm part of the QA/QC team here at Bethel Laboratories and Fortis Life Sciences. And I'm going to be talking to you today about antibody validation, why we do it, how we do it, and some of the specific implications for that validation in the immuno-oncology space. I'm going to actually start today by telling a little story about a protein called FOR. And FOR is a member of the JAK-STAT signaling cascade, and it was originally discovered early in the 90s, and then in the 2000s, it was implicated in cancer. And that implication was mediated primarily by an antibody called C20. And the Western blot in the bottom left-hand corner of this slide, you can see that the band indicated with the red arrow was thought to be FOR, and these are in multiple different cell lines, multiple different cancer cell lines. And that increase in expression was thought to have potential implications in cancer. And so they proceeded into clinical studies only to find out that C20 was in fact not binding FOR at all, but was in fact binding a heat shock protein known as HSP70, which had nothing at all to do with cancer. So obviously some front-end work trying to validate that antibody and really get a good feel for what it was doing, making sure it was binding to the right target protein, had a lot of downstream implications and a lot of loss, both in time and in some of the financial aspects as well. So the obvious question there is, well, how much money could you have potentially lost were a similar mistake made today? This image here is from 2014 and goes through a few different therapeutic areas and some of the costs associated with clinical studies in those areas. And you can see the immunology field that I've got highlighted. A phase 1 through phase 3 study totals off at almost $40 million total. And as you move into FDA review in phase 4, you're looking at upwards of $80 million. Potential patient costs with those studies through phase 3 comes up to almost $42,000. So obviously, front-end mistakes and a lack of validation could potentially result in hundreds of millions of dollars worth of losses for either the biotech companies working through these, labs working through these, licensing agencies working through these, and for patients as well. And this is particularly interesting in the IO space, because as you can see here, we've got a comparison of IO research and IO therapeutic and IO clinical research, and how that's changed some between the years 2017 and 2020. And you can see a handful of different therapy types over on the left. And you'll notice for each therapy type, we're seeing just massive growth, even between the years 2017 and 2020. It's estimated to be just over 230% growth just over this 3-year period. So combining some of the raw numbers that you're seeing here and some of the dollar amounts that you saw on that previous slide, you can only imagine some of the potential dollars that are at risk by generating some of these studies and then using reagents that have not been validated correctly. So that kind of begs the question, like, what is antibody validation and what do we mean by that? The FDA generally defines validation as the process of demonstrating through the use of specific laboratory investigations that the performance characteristics of an analytical method are suitable for its intended analytical use. So that's a very broad statement, I know. Within the antibody world, what you're really looking at there is to show that the antibodies that you're utilizing are both reproducible and they're specific. And that last statement there that I've got underlined about it being for its intended analytical use, I think really captures a lot of what antibody validation is about, because not only are you trying to ensure that your antibody is reproducible and is specific for what you're trying to do, it's also really important that the use of that antibody is consistent. And so whatever you're validating it for and what you're validating it to use it for is pretty consistent and very reproducible day to day. It's also important to keep in mind that there can, in some circumstances, be a difference between a validated antibody and a useful antibody. Now, I've got here a very clear-looking illustration of what a validated antibody may look like. This is an antibody against the target protein AHR on the far left, and we see a very nice, clean, single band. And then as we move from left to right, our Western blots are getting a little bit uglier. So this CAP1 antibody here in the middle we're seeing a very clear and obvious Cap1 Western blot with a nice clean band, but we're also seeing some off-target binding as well. And then as we move left to right, we're seeing this MLL1 antibody Western blot, where the blot looks a lot worse. We're seeing a significant amount of off-target binding, but we're still able to see this band. And so it becomes really important to be able to define what is validated, what is useful, is there a difference in those two things, and what are the implications of those potential differences. Antibody validation also can have some very unique challenges and some unique circumstances that you've got to be able to kind of work through and really understand the context of the biology of your target protein. So one of these particular challenges is the molecular weight pitfall. And by that I mean, when you go into a Western blot and something where you can very easily assess molecular weight, there are obviously a lot of resources available for you to be able to predict the molecular weight of that protein. Now, when the band your antibody is binding to migrates at a different molecular weight, you're left to answer the question, well, is this antibody on target? Here on the left, I've got ZBTB5, and this predicted molecular weight's about 75 kD. We see it here migrating in about 100 kD. On the right, we've got the Cap1 protein, again, migrating pretty close to 100 kD. Its predicted weight is closer to 88 kD. So you're sort of left to try and figure out and understand, hey, is this antibody on target? Why do I think this antibody is on target? And if it is on target, how do I prove that that is on target independent of just this molecular weight prediction? Something else that can be particularly tricky with antibodies in particular is sort of falsely associating the localization of antibody-derived signal on a tissue sample. So here we've got a non-Hodgkin's B-cell lymphoma kidney tissue that we've stained with an anti-EOMS antibody, and we know that EOMS is nuclear, and we're seeing very clear nuclear staining on this slide. Now, if this antibody were binding something off target that also happened to be a nuclear protein, you would have no way of differentiating that EOMS mediated signal from this off target binding. So again, you're left to answer some questions. How do you know that what you're seeing is on target? And if it is on target, how do you prove that to someone? One other potential problem is when you're seeing multiple bands showing up in a single lane. Oftentimes these can be associated with different isoforms or even tissue or cell line specific isoforms, or just some sort of post-translational modification of phosphorylation or glycosylation event. So in those scenarios, like for example here, as we're blotting with this anti-GATA3 antibody, we're seeing two very distinct bands in MCF7 cells compared to one distinct band in a DIRCAT T cell Western blot. And you're left with some questions here. How do I know what that second band is? Is it on target? Is it off target? And if it is on target, how do I prove that that is in fact on target? So this can create problems for both customers and for manufacturers and distributors of antibodies, because nobody wants to sell you something that's no good, and obviously nobody wants to buy a product that's not working well either. And so there's sort of a symbiotic relationship that can be really, really helpful as you're working through kind of characterizing and validating these in your own labs. From the manufacturer perspective, it's really important for us to be able to clearly communicate what we're doing, why we're doing it, and be able to talk to you in a way that's relevant to the work that you're doing. Oftentimes we'll do that through the provision of experimental results, experimental details. We've got a large body of data typically that's supporting a lot of these products. And when we're able to provide those to you as a mechanism for you to better understand your results, that can really help us in both directions. It's also important that the manufacturers work hard to define what that analytical use actually is. Oftentimes you'll see antibodies marketed for Western blot, for IHC, for ICC, for IP. Make sure that there's data that is supporting those analytical uses. If the manufacturer can't get it to work, but they're saying that it can work, that's usually a pretty good red flag for me that something's maybe not right, or that that thing hasn't been validated properly. I think I've alluded to this already, but it's really helpful for us to be able, or for manufacturers to be able to show as much data as possible, get as much information out there as you can, because the more information the customer ultimately has, the better off they're going to be in trying to figure out how to adapt and utilize that product for what it is that they're doing. And if and when a customer does reach back out to that manufacturer, make sure that there's more of a collaborative conversation there and a back and forth to try and figure out what's going on, and don't turn it into just a money back, return, whatever it is. You know, getting that product to work well is ultimately the goal for both sides. As the customer, you need to receive an antibody and be prepared to optimize the product for whatever it is that you're doing. Oftentimes you're going to receive a product that says, hey, we recommend this in a range, let's say from a 1 to 2,000 dilution to a 1 to 10,000. Don't always assume that one side or the other is going to work best for you. Be prepared to go ahead and validate that product, make sure that it's doing what you expect it to do, and then prepare for subsequent purchases of that same product to behave in a very similar way. And also going into it, make sure that you have a very clear understanding of what it is that you need. Don't buy a product for a Western blot when you don't need it to Western blot, or for an IHC when you don't want it to IHC. Make sure that what you're purchasing it for, that's what it's designed to do. Obviously, you want to make sure that the data that is being generated by the manufacturer supports the type of work that you want it to do. Check and see if there's citations where other researchers have used that product for what it is that you're trying to do. And if you continue to struggle, or if you just have some questions on the front end, don't hesitate to reach out to tech support. It's oftentimes that these tech support individuals are scientists who know really well what it is that you're trying to do and can help you sort of adapt and optimize your conditions to better suit those particular products. But still, this does present a very large challenge, and it's something that can be very fluid within the antibody world, just kind of at large. In 2016 and 2017, a group of researchers that ultimately became known as the International Working Group for Antibody Validation published a couple of articles where they were trying to sort of structure and organize the validation of antibodies and sort of standardize this across all of these different types of products. And what they ultimately proposed was a series of what they called pillars that should sort of support the research utilizing all these products. And these 5 pillars I've got listed over here on the right. These pillars included what they called orthogonal strategies, independent antibody strategies, genetic strategies, recombinant expression strategies, and capture mass spec strategies. We will talk about each of these in depth here in just a few slides, but I did want to kind of talk on a more 10,000 foot level before we moved into that degree of depth. You'll notice sort of in the antibody manufacturing community that we begin to adapt some of this language, and you'll see products and landing pages that are starting to allude to and refer to and sort of illustrate how these pillars are being used. We've got a Bethel Fortis landing page here on the left, Biotechne R&D, CST in the bottom right, and these are all sort of versions of this antibody pillar validation system that really sort of fits in well with what these companies do historically, and then are now being adapted to help better validate and better inform the customers who are actually purchasing these products. What's really interesting and really kind of cool about these pillars is that they have a what I would describe as an additive effect to a certain degree. So any one of these pillars by itself doesn't always have a ton of value. For example, if you've got a mass spec and you've got a Western blot and they look like they should, that's a good piece of data, but by itself you could kind of make some arguments to poke some holes in that. But when you start building, 2 or 3 of these pillars into sort of an overall validation strategy, the additive effects of multiple pillars can really do a lot more to support the validation of a given product. Now, to a point, that's true. When you start getting them above 3 and you start getting into like 4 and 5 pillars, it starts to become a little bit redundant, and the value gain by performing some of those other pillar validations, you're really not learning a lot more than what you already knew. It's also true that not every data set is going to contribute equally to your validation process. So, for example, CAP1 here in the top left, you're seeing a very ubiquitous expression pattern through all these different cell lines. you don't really know if that's on target or off target. It could very easily be an off target protein that just happens to express in every cell line. You don't really know. You don't really have any way to prove it. If you know that that Cap1 antibody has been validated, that Western blot creates some extra value for you. So this validation process isn't so much for you to learn that you can use it, or I guess the better way to say that would be validation teaches you that you can use something, the application data and some of these how to use something. It's also important to know that if someone is to validate an antibody, that doesn't necessarily make it useful for your work, it just makes it useful for theirs. And so it's important to remember that a manufacturer validation process should not replace your validation process in your lab, in your hands, in your conditions, in your cell culture conditions. There's a lot of novelty and a lot of variables that can really play into what's happening in your lab that are going to be different and independent and unique from how that product was validated by the manufacturer. So from the Bethel Fortis perspective, we've sort of adapted a little bit of a different approach to these pillars. So you'll notice that the top 4 pillars were actually the same, but we modified the capture mass spec pillar because we don't have a lot of mass spec capacity here at Bethel Fortis. So we replaced that capture mass spec with what we call a complementary assay and a biological strategies. And we're going to talk through those along with the other 6, starting with this next slide. So the first pillar that we're going to be discussing is what we refer to as the independent antibodies pillar. And this is basically defined as using 2 or more independent antibodies to generate similar results. And it's important to keep in mind that these antibodies need to be raised against different epitopes of that target protein. Now, historically at Bethel, we've used what we refer to as a reciprocal IP, where we do in fact use 2 different antibodies that were raised against 2 different epitopes. We will do immunoprecipitation pull downs with both of those antibodies and then blot with one of them. And in a situation like the one we have in the bottom left-hand corner here with AHR, if both antibodies are able to pull down the same target protein, we consider that to be a very strong argument for that binding our target protein. And it does fulfill the independent antibodies pillar, as it was 2 different antibodies being used to generate the same result. Over here on the right, this is a GATA3 stain of a tissue, and we'll notice that these are serial sections, and these 2 antibodies are staining an extraordinarily similar pattern in both slides. And so you would say here that data is the same, and that those 2 antibodies are performing the same and are binding to the same target protein, thus fulfilling this pillar. The next pillar is the complementary assays pillar, and this is the other half of the reciprocal IP, where we have 2 or more antibody-dependent techniques that are generating similar results. So with the reciprocal IP, you've got an immunoprecipitation and the Western blot of that IP that are generating similar results. This can also include other scenarios like the one that I've got illustrated at the bottom of this slide here, where we've got the same antibody binding well in a Western blot. This is a SOX10 antibody binding to SOX10 in an SK-MEL28 cell line. And you can see on the right here, this is an ICC image of antibody staining SK-MEL28 cells in culture. We're getting very strong signal, strong specific signal on both of these. We would say that this did, in fact, fulfill the complementary assays pillar. The third pillar that we're going to discuss is the orthogonal pillar, and this is where we use what we would call an antibody-independent technique to help verify antibody-dependent methods. And so the way this typically works is you'll pull something out of a mass spec scan or mRNA expression data, and then compare that to what you're seeing in a Western blot, IHC or ICC. And so in this particular example, we've got CD19 mRNA expression data from a database called Gene Visible. And you see that I've got 2 boxes in the bottom left-hand corner. These are boxing in the cell lines for Raji B cells and Dowdy B cells. And you can see on the right, we are getting very strong and specific binding to both of these cell lines. I've got those boxed in red as well. And this would allow us to sort of fulfill this orthogonal pillar during our validation process here. The fourth pillar is the biological characteristics pillar, and this is going to be a little bit more specific specific to the biology as it relates to your target protein. And these are more widely applicable where we've got some sort of phospho-specific antibody or some sort of protein that is expressed under certain biological conditions, but not other biological conditions. So here in the bottom left, I've got the induction of phosphorylation after addition of hydroxyurea to a cell culture line. We've got a phospho-CDK1 there in that top window, and you can see a very strong and clear correlation in the presence and absence of hydroxyurea. On the right, we've got a series of IPs where we have treated cells with etoposide, and that etoposide treatment induces phosphorylation, which we can detect with our phospho-specific CAP1 antibody there in the top corner. And we're also seeing the presence of total CAP1. You can see there in the reblot in the bottom of that window. And this, again, being able to sort of manipulate and use this biology to create the presence or absence of signal in a Western blot allows us to fulfill the biological characteristics pillar. The fifth pillar is what we would describe as the recombinant expression strategy. And this one's a little bit unique in that it doesn't stand alone particularly well, but it can have a very solid additive effect to the validation strategy for any given target. On the left, we are overexpressing MAF-A, MAF-B, and CMAF in a series of cell lines. And you'll notice that what we're trying to show here is specificity to show that our MAF-A antibody is not binding to MAF-B or CMAF. And you can see here in the top window that we are seeing very specific binding and that it is unique to MAF-A. This can also be really helpful in a scenario where whatever protein it is that you're looking for, it simply doesn't express well. And so you can recombinantly express those cells and then tag those, for example, on the right here with an anti-V5, or you can tag it with a V5 tag and then detect it with an anti-V5 antibody. And we can see staining in an ICC here on the far right, or even IPing after a pull down with an anti-V5 antibody here right next to it. So being able to kind of use those tags and some overexpressing proteins to help step into situations where the protein's important, but it's really difficult for us to find circumstances where those proteins are expressing at a sufficiently high level for detection natively. Finally, here in pillar 6, this is what we would refer to as the genetic strategy. And for all intents and purposes, you're going to know this as a knockout cell line. So basically here on the left, we have recombinantly removed ubiquinol-4's gene from a MDA-MB231 cell line. And we can see a very clear distinction between wild type and knockout cells. We've got very strong signal in the wild type. That signal is abrogated in the knockouts, and we can also correlate that in an ICC experiment as well. You see that over here on the right, where we're getting pretty good binding and staining in an ICC of wild type MDA and MB231s, where over here on the right, we've got knockout cells where that signal has been abrogated. So again, it's really important to keep in mind that a single one of these pillars, as a general rule, is not going to serve to say that this is a validated product in and of itself. Now, of these 6, the knockout genetic strategies pillar is probably the most capable of standing on its own. But typically we're going to kind of combine this with a few other types of studies and create more of a critical mass of data that is, very difficult to argue with for all intents and purposes. So prior to purchasing anything, it's really important to keep in mind, like, what do you need this antibody to be? And this kind of harkens back to that slide that I showed earlier with the kind of green-red gradient. Do you need something specific, or are you okay using something like this blot here on the right, where you can still see your target protein, but it's just not going to look as pretty? For some people, that's all you need. For others, you really need to be able to pin down something like this CAP1 blot here over on the left, where it's a nice, clean, sharp target band, nothing else on there. Again, keep in mind the question of how has this antibody been validated by the manufacturer, and make sure that you have a good feel for species specificity, what applications this thing has been validated for, and what cell lines has this been validated for. And finally, use your scientific training. Like, can you look through this data, and are you confident in this validation data, or do you look at this and you're concerned about what it is that you're seeing? Finally, after your purchase, make sure that you're using the manufacturer recommendations as a guideline for how you're utilizing this product. Are you building in some of their positive controls? Are you sticking to their validated species? Are you sticking to their validated applications? Don't be afraid to optimize this product. Find the conditions that work for you. Do be aware, especially with polyclonal products, that as these antibodies age or as lots change, make sure that you revalidate subsequent lots. Make sure that you know what the implications of those lot changes are, how it affects your work, and ultimately what you need to do to adjust for that. And finally, again, don't be afraid to reach out to us. Most of the time, us antibody manufacturers, all of our colleagues in this field, we like talking about these products. We take a lot of pride in the manufacturing and testing of these products, and frankly, they're fun for us to talk about, especially with people who are passionate about this work, what we're doing, and really want to dig into and understand the science behind these antibodies.